Hello and welcome to Lecture 11, Cellular Respiration Part 2. Today we're talking about the oxidative phosphorylation step of cellular respiration. Let's recap from the last lecture. So we talked about cellular respiration at that point as well. Here we talked about the fact that cellular respiration breaks down sugar to produce ATP and carbon dioxide and that it requires oxygen. We said that redox reactions are coupled and move electrons between reaction partners. We said that in cells, electrons can be moved to electron carriers, which can help transfer electrons and hydrogen. And then we said that cellular respiration involves 4 major steps. And we talked about glycolysis, pyruvate oxidation, the citric acid cycle, but not oxidative phosphorylation yet. That's for today. We said that in each step, ATP, NADPH, and carbon dioxide are produced, and oxygen is used in the last step. And then we looked at exactly which of these compounds are used in which of the steps. So you should memorize that part. And then citric acid cycle is used in many metabolic pathways, not just in cellular respiration. Today we're going to see one of those examples at the end of the lecture. Okay, so I know that that was a lot last time and that the biochemistry is sometimes pretty daunting for somebody who hasn't looked at many of these chemical structures at all. And so what I want to do is I want to go back to the overview. I want to remind you of why we're doing this, what's going on here on the big picture, and then we'll go back into the mitochondria essentially to look at the last part of cellular respiration. So this is a slide that I showed you last time, and I said cellular respiration involves 3 or 4 major steps, and these steps are listed here on the side, and it says 3 or 4 because pyruvate oxidation is sometimes considered as part of glycolysis. But it's an important step, and so therefore it gets its own stage here in this list. And then remember that this is a sort of a pathway overview picture so that you can see where you are when we zoom in to really understand what's going on the molecular level. We had talked about glycolysis, we had talked about ATP production, we talked about the end point of glycolysis from glucose being pyruvate then being oxidized in the step where it's moved into the mitochondrion. We said carbon dioxide comes off there. Acetyl-CoA was the product that was produced at that point, which is then fed into the citric acid cycle. There again, carbon dioxide comes off, ATP is produced in substrate level phosphorylation, just like here. And then what is also happening in addition to that is that electron carriers are picking up electrons with hydrogens, and they then transfer that to the electron transport chain. And this part here we haven't discussed. We're going to discuss that today. We hadn't looked at these 2 different steps here. Today, what we're going to do at the end of the lecture is we're also going to very briefly look at fatty acids and how they are metabolized in this pathway and how they end up as acetyl-CoA essentially, and then the rest of the metabolism of fatty acids is through the citric acid cycle and electron transport chain. Okay, so we briefly talked about the mitochondrion last time, and we had talked more about it in previous lectures. It's an organelle in eukaryotic cells. It's found both in plants and animals. It's required to produce lots of ATP. And so this here is a blow-up of this mitochondrion, and specifically what part of it is blown up here is that you see that there are 2 membranes, right? There's the inner membrane, it's all this highly folded, and then the outer membrane that's just the envelope around it. So that's the outer membrane here and the inner membrane here, and that is critical in the process of electron transport chain and what's called chemiosmosis, where that together you have oxidative phosphorylation, you're producing a lot of ATP, and I'll show you today how that happens. But what this figure shows you here was how pyruvate was added into the mitochondrion, and then all the other steps from there on out occur in the mitochondrion. Okay, I want to show you another overview. There's never enough of these overviews. It's a very complicated process, and it's good to remember where things happen in the cell. And so what this figure shows you is this structure here that kind of looks like a brain, but it's supposed to be mitochondria. And you see that it has the two membranes inside the inner membrane space. So this is the mitochondrion. These steps all occur inside the mitochondrion. These steps out here occur in the cytoplasm. Right, so glycolysis from glucose to pyruvate, producing electron carriers via NADH, producing some ATP. Pyruvate goes into the mitochondrion. Here is where the citric acid cycle occurs. More electron carriers are produced, and then those, together with these electron carriers, are fed into what we're discussing today, the oxidative phosphorylation steps, electron transport chain, and chemiosmosis. And then that produces a huge amount of ATP, and that is called oxidative phosphorylation. This whole step here, this whole process is called oxidative phosphorylation. Okay, so electron transport chain. So you have all this FAD and FADH2 and NADH, right? Those were the electron carriers that were produced during the previous steps in the citric acid cycle. So what are we going to do with these electron carriers, and how are we going to produce ATP out of those? That is what this figure shows you. And your book has another figure, and I'll show you that as well. I really prefer this because it's simpler, but the other one is, a little bit more accurate because it has better structures. This just has blobs, but the blobs are good enough for right now to get me started. So this here is the inner membrane. See how it says inner membrane here? And then the mitochondrion has the outer membrane, but it also has the intermembrane space between the inner membrane and the outer membrane. So between those two membranes is the intermembrane space. And that is this dark shaded orange. That's the intermembrane space. You don't see the other membrane, right? This is one inner membrane. Out here somewhere is the outer membrane. And then inside that is the mitochondrial matrix. That's the very inside of the mitochondrion. If we look back here, the matrix is that stuff that's in the very center. The intermembrane space is the part between the two membranes, and then of course you see the two membranes. So all this NADH is moving into the mitochondrial matrix, and there we have two processes occurring. One that's called the electron transport chain, and that is all of this here. And then the second part is called chemiosmosis, and that's this part here. Chemiosmosis is where actually ATP is produced. And chemiosmosis is a process that essentially uses a membrane transporter. We talked about membrane transporters in class before. This is a specific membrane transporter. We had talked about active transport and how ATP is used to produce, for example, a gradient or pick up something against its concentration gradient. This membrane protein can use an electrochemical gradient, and that electrochemical gradient, for example, through the formation of this H+ gradient difference between the intermembrane space and the mitochondrial matrix, that can be used to power this this membrane transporter. And it's a very special membrane transporter. It's called ATP synthase, synthesizing, so it produces ATP. And it does that here in the mitochondrial matrix. We'll come back to that at the end, but that's where it all goes. In the meantime, we have the electron transport chain, and the main point of the electron transport chain is to set up an electrochemical gradient. So if you want to think about this in two steps, this part sets up an electrochemical gradient, and the electrochemical gradient is then used from in this part where the ATP synthase takes all these hydrogens. They run through this membrane protein, and as you see later, it's sort of a little molecular motor on there like a turbine, and these hydrogen ions, these protons, they spin through this turbine, and that turbine produces kinetic energy, and that kinetic energy is transferred onto the ADP, which makes ATP, and that's, you know, that's the whole point of the entire pathway of cellular respiration. So how is this electrochemical gradient set up? That's what's happening over here. And how does that happen? Well, it happens because the NADH carries electrons. Right? We looked at the molecular structure last time. We saw how it can take an electron to become from NAD plus to NADH. So it picks up an electron. Picking up electrons is a reduction. It also picks up a hydrogen, like a proton. And together, the hydrogen, it reduces the NAD plus to NADH, right? So we have reduced NADH electron carriers. Those are the ones that come into the mitochondrial matrix. And so since they are in the reduced state, they can now give off their electrons in a redox reaction, right? We talked about redox reactions last time, where you have a coupling of reduction reactions and oxidation reactions. This guy is carrying electrons, so this guy can be oxidized, and as it's being oxidized to NAD+, the electrons have to go somewhere, right? The electrons, since this is a coupled reaction, the electrons have to go somewhere, and they have to reduce something. And that's what they're doing. This blob, essentially, is what they're reducing. Well, that's a very complicated structure, and so therefore this book only shows you, a very reduced representation of that. But essentially what's happening here is a redox reaction where NADH is oxidized to NAD+, and protein complex 1 is reduced by these electrons. In redox reactions, we said energy can be liberated, and that energy that can be liberated can do work. And the work that it does in this case is the pumping of hydrogen ions from the mitochondrial matrix into the intermembrane space. So the redox reaction here is coupled with the pumping of hydrogens from the mitochondrial matrix into the intermembrane space. That is starting to produce for us a hydrogen gradient. So now the electrons are sitting here in protein complex 1, and they have to go somewhere. So they're going in a second redox reaction to the next compound along this chain, this long chain. And that's why it's called an electron transport chain, because the electrons are transported along this chain from the protein complex 1 to 2 to 3 to 4. There are these intermediates, electron carriers that shuttle the electrons from one protein complex to another. And in each step, a little bit of energy is liberated, and there are 3 places where that liberation of energy is big enough to drive the transport of protons from the mitochondrial matrix into the intermembrane space. So successive reactions, redox reactions, some liberate energy. That energy is used for the pumping of hydrogen ions. At some point, these electrons still have to go somewhere, right? So they're carrying less and less energy. The substances that are reduced, that now have the electrons, carry less and less energy. We looked at this gradient, this, you could call it a gradient, this loss of successive energy steps from step to step, successively from a higher to a lower 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 level of energy content. And that is what's happening here. So these electrons that are reducing a certain compound together carry a certain amount of energy, but with each step, they're losing some of that energy. And at the end, there's hardly any energy left, but the energy was used for the pumping of these 3 protons out through the inner membrane. And at the very end, with the last breath, if you will, of course there's no breath here, but with the last bit of energy that these electrons have, they are now reducing oxygen with the help of 2 protons to a water molecule. So oxygen is reduced to water. That's the last step that these electrons are doing. And then they're finished. In the meantime, through all these redox reactions, they have produced an electrochemical gradient, and in the end they have produced water. That is the water that you evaporate, that you breathe out as a result of cellular respiration. That's the water here. And this oxygen here is the reason for why the whole thing is requiring oxygen. So you can only do cellular respiration, of course, which means breathing, with oxygen. So this step here is not working unless there's oxygen. And if there is no oxygen left, then these electrons can no longer move from one step to the next, because it's basically backed up. It's clogged at the end. If the electrons are not going anywhere, then they can't, then there are no more electrons liberated from the NADH. So unless they can go to their final acceptor, which is oxygen, they get stuck. If there's oxygen, then successively the hydrogens with the electrons come off. The electrons are reducing the next blob down, the next protein complex down, and at the end they reduce oxygen to produce water. So here we have our electron transport chain. Here we have our electrochemical gradient, and then in the end we can use that electrochemical gradient to power this protein, which is called ATP synthase, that makes ATP. So now let me show you what your book has. And the first thing I need to mention, point out to you, is that this is upside down compared to the previous picture. So here the inner membrane space is on the bottom and the mitochondrial matrix is on the top. Of course, that doesn't matter where it is, right? The mitochondrion itself is looks like this. Whether you look here, whether you look there, that's upside down or inside out or right side up, whichever way you want to look at it. Just make sure that if you use this figure versus this figure to understand what's going on, that you realize that difference. So why am I showing you this? Well, I'm showing you this because your book has it, and so you'll be looking at it anyway. I find it a little more complicated, but really you can walk through it. We can walk through it together right now quickly. The nice thing about this is that you see the three-dimensionality of these complexes, and you see a little bit how that three-dimensionality of these complexes is used for the movement of hydrogen through these pores, through these channels, through these parts of the proteins that allow the movement of something through them, which the membrane itself would not allow. So you can see NADH, you can see FADH2 being oxidized here. You see the pumping of the hydrogen here in 3 different places. You can see, and that's also quite nicely done here in this figure, the movement of the electrons, right? So that's what's called the electron transport chain, of course, how it is moving from one protein to the next, each time losing a little bit of energy, and at the end, taking oxygen together with 4 hydrogen ions, protons, to produce water. So that's pretty nice. And then it also shows you here in this form the individual steps. So let's look at these individual steps, because there are some parts in between that the other figure that I didn't discuss in the other figure. So there are these intermediates, coenzyme Q, which are reduced to coenzyme QH2, and then they transfer the electrons from complexes 1 and 2 to complex 3. So that's what's happening here. These two are gathering the electrons from these 2 complexes, deliver them here. While, of course, in complexes 1 and 2, the electrons are harvested from NADH and FADH2. So then this little guy here in between, cytochrome C, moves to complex 4, where oxygen is reduced to form water. And when I say it moves to complex 4, it really means it takes the electrons from complex 3 and moves them to complex 4. Redox reaction, it's reducing complex 4, giving off the electron, and then in the end you have the formation of water. So the transport of electrons in complexes 1, 3, and 4 is coupled with the transport of protons across the inner membrane from the mitochondrial matrix to the intermembrane space. I told you that already previously. So that's the pumping that's discussed here. And then we spoke about this. ATP synthase uses the electrochemical proton gradient to drive the synthesis of ATP. Here's ATP produced. That's the three-dimensional structure. Hydrogen moves through it. And the way that this looks, let's look at this little bit as a cartoon. You can see as the hydrogen moves back through, driven by the concentration gradient, right, by the electrochemical gradient, it's like a rotor with rotor blades. It moves through, and it does that because it follows its concentration gradient. That pushes these blades, these molecular blades. This is all proteins, so parts of the protein structure, the three-dimensional structure of the proteins, that allows the change of the energy of these hydrogen ions rushing through to be turned into kinetic energy, and that twists something. And as that's twisting, it can grab onto an inorganic phosphate. That's what that little I stands for, inorganic phosphate, and ADP, stick them together, costs energy, but that energy is delivered by the movement of the hydrogen through that mill. So it's sort of like a water mill, if you will, on a molecular scale. So that is then producing the ATP, and this is happening in the matrix from the intermembrane space. Okay, so there was a lot of stuff. What is it that you need to know? You need to know quite a few things about this. So let's go through this, and then you can come back to this particular slide and see if you understood each of the parts. So oxidative phosphorylation is comprised of two steps, electron transport chain and chemiosmosis, which is the ATP synthesis. Electron transporters are oxidized while delivering electrons to the intermembrane protein complexes. Those are the blobs that I showed you, 1, 2, 3, and 4, and the two intermediates in between. And they're reduced, of course, right? They're reduced because hydrogens, electrons are given to the next. So the electrons here are important, right? So that's the reduction. So they're reduced. And then in successive redox reactions, the electrons are handed down from one complex to the next. Eventually, they react with the two hydrogens and molecular oxygen to form two water molecules. So that was the final electron acceptor. Overall, this produces 28 ATPs. There's some water that's produced, and it requires oxygen, right? This process requires oxygen. That's why it's called oxidative phosphorylation. I already said this. Oxygen is the final electron acceptor in the transport chain. The process takes place in the inner mitochondrial membrane. The redox reactions along the membrane complexes use the liberated energy to pump the hydrogen into the intermembrane space, creating that electrochemical gradient, and then that gradient is used to power the membrane protein ATP synthase, which produces the ATP. So look at this. This is what I would like you to remember about the steps. Okay, let's take a look here at the energy balance overall. So remember, this is the entire pathway. Glucose is broken down in glycolysis to pyruvate. Remember that there was a little bit of energy that was required to jumpstart that process, and then there's some ATP that's generated. Overall, there's a little bit more energy that comes out than goes in. There's also some electron carriers produced. Those then get fed into the inner mitochondrial space. So you see this is our mitochondrion here. These white lines here are the inner mitochondrial membrane itself. So pyruvate was produced. Pyruvate is oxidized as it's entering into the mitochondrion, and then some NADH is produced in that step as well. Through the citric acid cycle, or the TCA cycle, or the Krebs cycle, all different names for the same thing, electron carriers are produced, a little bit of ATP in substrate level phosphorylation, but then the major phosphorylation is the oxidative phosphorylation, which happens in the electron transport chain. 28 ATPs, total of 32 ATPs then. So you can see that almost all, right, 28 out of 32 of the ATPs that are produced in the electron transport chain, and they require oxygen. No oxygen, no 28 ATPs here. You can, without oxygen, generate this part, but you cannot then use these electron carriers for the electron transport chain, and you don't get these 28 ATPs. So this table here, also from your book, shows you the individual steps, what you get here in terms of NADH equivalents, and 2 NADHs produce more or less 5 ATPs. So that's not quite as easy to see because it depends on how many hydrogens are pumped, and then they have to go through that ATP synthase. So this is sort of the conversion rate, if you will. Here's your sum. You can take a look at the total, 32 in the end. That is oxidative phosphorylation, and that is the end of our pathway here, where we are breaking down glucose with the use of oxygen to produce ATP. Now, what if there is no oxygen? Some organisms can live with oxygen, going through all of these steps, or without oxygen, going through these steps, but then not going through the last set of steps. And there are some quite well-known organisms that do that. They can not use oxygen, or they can use oxygen, but they can also live without oxygen. So what happens when they do not have oxygen? And that is what I call here life without oxygen. So, and that process is called fermentation. You've heard of fermentation. Fermentation produces alcohol, and it produces lactic acid, for example. There's other things that can be produced through certain bacteria that do these steps, especially the steps here at the end, are specific to specific species of bacteria, and or yeast in this case. So yeast, for example, is producing is producing alcohol without oxygen. And so how does it do that? You already know most of these steps of fermentation because all of this here is glycolysis. So it uses glycolysis just like a cell would that uses oxygen in the end for oxidative phosphorylation. Well, it doesn't have oxygen, so it can't do oxidative phosphorylation, but it has pyruvate here. Well, what's it going to do with it? It's really stuck. It needs to get rid of that. And so yeast cells excrete their waste not as pyruvate, but they take off the carbon dioxide in a very similar process to the pyruvate oxidation that we saw as it's entering into the mitochondrion for oxidative phosphorylation. It's producing acetaldehyde. So see the acete here? We had acetyl-CoA. Well, in this case, we're making acetaldehyde, no CoA stuck on. Carbon dioxides come off, and then this requires these NADHs that were produced. So it now needs these hydrogens back. So it takes those and it sticks them on the acetaldehyde, and that allows it to produce ethanol. Now it has NAD plus again, which it needs for glycolysis to work. So it's making NADH. Can't use NADH anyway because it has no oxygen, so it might as well use the hydrogens for producing the waste product that it can excrete, which is alcohol. So then alcohol gets excreted into the medium. Yeast lives in some sort of a medium, liquid medium. It excretes that. The waste is gone. Now it can break down more sugar. And so that is what alcohol fermentation is. So it basically, the alcohol that is produced is excreted out of the cells is then what's in the in the medium, and we might call that medium beer or wine, where that alcohol then accumulates as yeast breaks down more and more sugar. At some point, there's so much alcohol in the surrounding medium that kills the yeast. That's why yeast cannot produce a higher concentrated alcohol than a certain percentage. So wine, for example, the fermentation will stop when wine has reached 12% or so of alcohol. It kills the yeast. Fermentation stops. That's the reason for why that's the maximum of alcohol that you can get through the fermentation process. Another fermentation process is lactic acid fermentation. Here you produce lactic acid as a waste product. And that's, of course, in the eye of the beholder, right? So it's waste to the lactic bacteria that are producing this. They want to get rid of it so that they can break down more sugar, so that they can get that electron acceptor back to the oxidized state, which is required for glycolysis to proceed. So pyruvate here is excreted as lactic acid. And so that is what happens when you ferment milk. Milk contains milk sugar. That's why it's slightly sweet. That also has the that then produces the lactic acid in the end. And so that's the fermentation that occurs during lactic acid fermentation. So what do you need to know about fermentation? You need to know that there are different kinds. I just showed you the two examples. But what's common to all of them is that it allows ATP production without oxygen. So you have no oxygen, you can still do it. You can still do ATP production, but you can nearly get as much, right? It's a lot less effective, lot less efficient than oxidative phosphorylation, produces 2 versus 32. It uses glycolysis, right? And it produces a waste product. It could be, that waste product could be alcohol or lactic acid. So essentially, fermentation means just using glycolysis and producing some sort of waste product. All right, so that is fermentation. I had told you that I wanted to look at some other pathways that use the TCA cycle. And so I had said, I pointed out at the very beginning, let me point that out again. I pointed out that here we saw glucose and glycolysis producing acetyl-CoA, but that fatty acids and amino acids can also be broken down, and they can also produce acetyl-CoA, and then they can be fed into the citric acid cycle. So what that means is that when we look at fatty acid breakdown, remember that fatty acids have these long carbon-hydrogen chains, and they can be broken down by certain enzymes in steps of 2. So they take off these last 2 carbons, and they essentially make acetyl-CoA from them. So see how this is essentially acetyl-CoA. This here is acetyl-CoA after the addition of CoA, and that's the acetyl group. So 2 carbons with the oxygen, and then that comes off in a reaction where that oxygen is with this group with the oxygen is transferred to the CoA, and where that then can be fed into the citric acid cycle. And in this process of shortening the chain bit by bit, you also get some FADH+, FADH2, and some NADH, and those can be fed into the electron transport chain. So that's a very brief discussion of beta-oxidation. What I want you to remember of beta-oxidation is that it's the name of the metabolic pathway in which fats are broken down, that the fatty acid chains are successively shortened in increments of 2 bits. These are the 2 bits. See, now it's 2 bits shorter. That's the next step. And then so it basically eats itself backwards until the chain is gone. The 2C pieces, the 2 carbon pieces, because it has 2 carbons, right? 1, 2 carbons. These 2 carbon pieces become acetyl-CoA, which is fed into the TCA cycle. And during the shortening process, these electron carriers are also produced, and they are fed into the ETC, in the electron transport chain. So that is what you should know about beta-oxidation. Okay, give me a few more minutes, and I want to say just a few words about how this whole pathway of cellular respiration is regulated, because it's not going on continuously all the time, which would be pretty wasteful under conditions where the cell doesn't need any more energy. Right? Once it has a lot of energy, it doesn't need to maintain that entire pathway at full force. And so there are steps that are sort of putting the brakes on or the accelerator down, depending on what is required. And so here in this figure, you see the steps again. You see glycolysis, you see the entering into the mitochondria, and you see the citric acid cycle, electron transport chain, and you see here how certain steps are inhibited with these red pathways and the minus signs, and where the pathway can be activated. And so what activates the pathway is ADP. Of course, the cell has a lot of ADP when it doesn't have a lot of ATP, because every time you break down ATP, what you have is energy, which is great. That's why you want ATP in the first place. But the leftover product is inorganic phosphate and ADP. And so a lot of ADP means not a lot of potential energy available for the cell. Likewise, a lot of ATP means the cell is flush with energy equivalents. It could do all kinds of things with it. It doesn't really need more right now. So that means that if it can signal to the cell to itself, stop with this pathway for a while, we're good, then that would be a nice way to give feedback, right? So that's exactly what it does. It gives feedback here by inhibiting certain steps within these pathways. There are multiple steps that can be inhibited, or likewise activated if ADP is around. So how do activation or inhibition work? Well, we talked about that 2 times ago. If you remember, we talked about enzymes and how enzymes can be activated, we said allosterically, and I'll show you again that word and what that means, but how enzymes can be, how they can become more active or they can become less active. And that is because something binds to their 3-dimensional structure, and that then inhibits or changes their active site. Let me show you a picture of that. So if this is an enzyme, example, phosphofructokinase 1. You don't need to know that name. You don't need to remember that. That is an enzyme that's required during glycolysis. So just as an example, this site here, this 3-dimensional site, it's 3-dimensional, it's a blob. It has a certain area where it can bind to ADP. And if something doesn't bind to the enzyme in its active site where the action occurs of whatever it's doing, then that's called allosteric binding. So binds somewhere else and can change the 3-dimensional structure through this binding of the active site. So ADP binds allosterically somewhere else to this enzyme, changes the active site, and when the active site is changed, in this case, it becomes more active. It becomes just right so that it can grab the substrate, which in this case is fructose 6-phosphate, and produce the next step down the road, which is fructose 1,6-bisphosphate. And so then glycolysis is turned on. You see the dark green arrow indicating that. So when ATP levels are low, this enzyme is activated, allowing glycolysis to continue. But when ATP is high or citrate levels are high, then they will bind here allosterically. They will inhibit this, and then glycolysis is dimmed out. You see that this is no longer dark green, suggesting that it's now no longer active. So in short, an example of allosteric enzymatic feedback inhibition, which tells you more about enzymatic interactions in general, but here we have a nice example for how that works with respect to the pathway that we just discussed. Specific enzymes are inhibited or activated, and this can occur here during times when ATP is low or when ATP is high, which is a nice feedback for the cell to either ramp up or turn down the pathways that lead to the formation of ATP. All right, so let's sum this up. We had quite a bit of stuff to discuss in these last 2 lectures. So today we talked about the fact that cellular respiration breaks down sugar and produces ATP and carbon dioxide, and it requires oxygen. Finally, this first sentence is exactly what we also discussed last time already. But today we talked more about this process. Oxidative phosphorylation occurs along the inner mitochondrial membrane. The oxidative phosphorylation is comprised of the electron transport chain and then the chemiosmosis. Those were the 2 parts. During the electron transport chain, electrons are passed along a series of membrane proteins. Each step is a redox reaction, and the last step delivers the electrons to oxygen to produce the water. Hydrogen gets added with the hydrogen ions, so in this case protons, right? No electrons. They take the electrons plus the protons with the oxygen, and that produces the water. During the redox reaction, the electron transport chain pumps hydrogens through the mitochondrial matrix. That creates the electrochemical gradient. That gradient then powers ATP synthase. And that is what oxidative phosphorylation is. And then also we discussed fermentation. We talked about beta-oxidation. I showed you these two examples because I wanted to show you how parts can be used for other metabolic processes that aren't cellular respiration. But here we have an example of glycolysis being used as the main step of fermentation. And here we have an example of beta-oxidation using the TCA cycle and oxidative phosphorylation to break down fats. And then we have cellular respiration regulation through enzymatic activation or inhibition through feedback. So look at this again, read these sentences one by one, see if you understand what they mean. If not, go back to the lecture, look at the slides, see if that helps, and if not, you can always ask me, of course. All right, that's it for today. I'll see you in class.